All right, let's go ahead and get started. Again, I am Bob Kemerait. I don't care if you follow me on Twitter or not. You know, I'm struggling today. What does not remembering how to tie your tie mean? So everything else is inconsequential. So you're not going to hurt my feelings. But if you do want to follow me on Twitter, I promise you on Twitter that I do not post anything other than things that I'm seeing in the field. Or, for example, Eddie Beasley in the back will send me pictures of things that he's seeing, and I post them which are of interest. So if you want to see what we're seeing, the main thing, Seth, is it's a segue into county agency consultant, okay? And so here's what I want to talk about today. Here's what I'm going to talk about is the La Nina we're having, opportunities for nematode control, bacterial blight just a little bit. Okay, bacterial blight wasn't a major problem last year, but we have to continue talking about it. A little bit about Fusarium Wilt. When I started in 2000, I didn't even know what these two things were, but they're really driving in some ways now. Foliar disease management becomes increasingly important. And I had a meeting yesterday in Tift County, and the growers were looking for ways that they could cut input costs, and they were suggesting, do we really need to spray for foliar disease? Seth McAllister did a trial last year in Terrell County. We'll show a little bit of the data. The answer is you don't have to spray for foliar disease. You can make good cotton without spraying for foliar disease, but you make more cotton and more money at times when you do, okay? And I'll let Sudeep talk about the cotton leaf roll dwarf virus. Here's my world, okay? It hasn't been too long ago when Bob talked it was going to be seedling diseases and nematodes, okay? What's happened since then? What's happened since 2000, Rick? New varieties, new nematicides, new diseases. So it really has continued to evolve. It's not because of me, it's just because what we have to do in order to produce better cotton. And if you look at our disease loss estimates, and I know you can't read this, but our estimates from Georgia are the lion's share. The lion's share of losses, good morning, Sudeep. The lion's share of losses in my world are going to be from nematodes. And across Georgia, it's primarily going to be root knot nematodes. But we do have reniform as well. We do have sting nematodes. Mark, as you know, in your area, sting nematodes can be a huge problem. Okay, but we'll talk about that. But what's interesting is, even though the losses overall compared to nematodes, target spot and aerial mildew certainly are providing an opportunity, a cost, but also an opportunity to make more cotton. You're going to have to spend on it, but you're going to make more cotton. Okay? Let's talk quickly about the La Nina. The good news is in Tifton, Georgia, Saturday morning is predicted to be 24 degrees. Okay? We have not had much of a winter yet. Yes, we've had some cold weather. A La Nina is characterized in the southeast by warmer and drier. That doesn't mean it's always warmer and drier. It just means that on average it's a warmer, drier year. So why does that matter? This is not cotton, this is corn. Okay? Eddie Beasley in the back sent me this picture. Hey, you were in Tift County, what, about 5 miles from here, right? Yeah. About 5 miles from here. That's corn on January 24th. Volunteer corn from seed that had spilled, okay? And it's looking a little haggard there now, but it was still alive. And it's covered with a new disease called tar spot. New disease to us, the major disease in the Midwest. And the question is, not the question, the problem is if we're on January the 24th and we still got corn that's at least surviving and continuing to produce the kind of spores, what's that mean for going into 2022? Neutral phase, or we're in a La Nina phase, not neutral. Warmer than normal spring, warmer than normal winter, warmer, drier. What's it going to do primarily in my world on cotton is going to be what you're going to do for nematodes, okay? And these are what the biggest problem is this. The 21-day life cycle, root knot nematodes, from the time it's an egg to the time it is a female producing eggs, is about 21 days under optimum conditions. When soil conditions get below 65 degrees Fahrenheit, which they are, understand when they get below 65 degrees Fahrenheit, those nematodes quit feeding. And below 50 degrees, they pretty much become sedentary. They pretty much become not doing much at all. And probably our soils right now are about that or below that. So right now they're not doing much. The problem is this. 14th of January, within a 5-minute walk of here, there was cotton blooming. In Mitchell County, if you look down the road, you can't see it very well, but all of the cotton's regrowing. So hopefully we've frozen some of that back. The problem is the winter we haven't had yet And the warming conditions we might have after a cold front mean that we may still have some living host out there. And as those soils warm up, it's a problem for being a source of food for the nematodes. Okay? My number one concern over the winter we haven't had, and it may get cold and it will get cold, John, this Saturday. I know that. But what's going to happen after? How quickly is it going to warm back up? We may have, if nothing else, we've continued to have nematodes active and diseases active long after they should have been put to bed. I look at our winter months in Georgia as being, if I took my phone out right now and I shut it off, if I took my computer and shut it down and unplugged it from the wall, that is what winter months should be for cotton growers in Georgia. It's a reset button. What is this? What is this? This is 2021. This is 2021 that doesn't care about a calendar, so we really need cold temperatures. Okay?
Will a 24 degrees kill? It will not actually kill the nematode, really?
No, it will not kill the nematodes. It will send when the soil temperatures get below 50 degrees, which they may be right now, the nematodes are not killed, they're just not doing much.
What would it take to kill? I mean,
we will not, it would take 19, you know, it's not only the temperature, it's the prolonged period of the temperature. So if we dip down to 24 degrees for an hour, then go back up during the day, we're talking about to kill or actually really have an impact, we need to be 24 degrees for 8 hours or so. You know, it's not only the intensity of the cold, it's the duration of the cold. Okay? The other thing about this, and Sudeep will talk about it, I'm sure, is what does this mean? If we have a living cotton plants out there, if they don't survive, if the roots survive, you kill back the foliage, they leaf back out, is it a reservoir for other diseases like the virus? Okay? Main point and the first point out of 7 steps today is just recognize whatever happens this winter, farmers don't farm from March till November. Your farming operation goes year-round. And what's happening right now and what may happen as far as the spring, is it cooler, wetter, warmer, drier, it will impact not only the nematodes but the other diseases we face. And so you have to make the right decisions on that. Okay? Talk about opportunity. There are significant things, if you believe me, if you agree that nematodes are the biggest problem in my wheelhouse that I face, there's significant opportunity that happens only one time in the season. And that one time is before you close your furrow. Once the furrow is closed, that opportunity is gone. I parked right next to you. You know your truck is hard to miss. I was careful. I didn't bump into it or anything. Okay? Well, I knew your truck. Okay? The opportunity is you get one chance for it. You go from there. Okay? Biggest thing in my wheelhouse is nematodes. Primarily the root knot nematode. Left-hand side is Screven County, Georgia. Right-hand side is Colquitt County, Georgia. From coast to coast, from top of the state to the bottom of the state, if you grow cotton in the state of Georgia, you have to be aware of root knot nematode. That's not the only ones, but one we have to be concerned with. And this slide, if you remember nothing else from my talk, you don't care, you go right to Sudeep and you hear his great talk, you don't remember anything else I said, remember this slide. Bullock County, 2017. This grower chose to plant what he knew to be a high-yielding variety and gambled that the nematodes would not affect him. He went with a low rate of aglogic, 3.5 pounds, a thrips rate, and he went with a successful variety, highly yield potential, but no root knot nematode resistance. And he made all those decisions in early May or late April. And for the rest of the season, he had to watch because there were no answers after that. If you remember nothing else, the decisions you make as far as the variety you plant and what nematicide you use or you don't use are going to be everything you can do during the season for nematodes and for bacterial blight, whatever that may matter or not. Okay? And this grower had to watch. That's not the grower, by the way. The grower wouldn't come out. He sent somebody else. Right? But that's what happens. Okay? Just to reiterate, here's Georgia. This is southern root knot nematodes. Red is covered up. Orange is almost covered up. Yellow is getting there too. Okay? You can see wherever we are in the state of Georgia, you're going to have root knot nematodes. But you can also see the reniform nematodes at the top. And along the fall line especially, you can see we got plenty of reniform nematodes here. They're just not as plentiful. If you grow cotton in Georgia, you have to worry about the nematodes. Okay? Why do we keep talking about nematodes and resistant varieties? The first time I tested resistant variety was a Phytogen 367 back in 2010. And what we learned in 2010 was if you plant a root knot nematode resistant variety, you have less damage to the roots and you don't build nematode populations up. So why keep talking about it? I can tell you in 2022, you plant a root knot nematode reniform resistant variety, you're going to have less damage and you're not going to build the nematodes up. So why talk about it? Because we continue, as we improve the cotton varieties, John, we continue to bring those same improvements into the resistant varieties. The yield potential, why did we plant much 367? Because of yield potential. But the new varieties continue to have an increase in yield potential, which is competitive, increasingly competitive with our best varieties. And the last reason is, if you have the chance in 2022 to plant Phytogen 443 or Delta Pine 2141, it comes with root knot and reniform resistance. If I was growing cotton in Tifton, Georgia, Tift County, what would my question be? Does the reniform resistance, I don't need it. Does it cost me anything in terms of performance for root knot resistance? Does it cost me anything as far as yield goes? Because I don't really need the reniform. Here, tomorrow I'm going to be in Jay, Florida. They don't care about the root knot resistance. All they care about is the reniform. They're going to ask the same questions. That's why we keep talking about it. Okay? This is from 2021. I don't have all the information for 2022, but I can assure you that most of this will be very similar. Here's root knot nematode resistant varieties. Phytogen varieties in red are some of the newer varieties. We still got Stoneville 5600. We got 1747 out there. Okay? You get one chance to select. You get one chance to select the variety you're going to plant in a field unless you have to replant. The point I want to make is if you are planting these varieties, the main point is that with root knot nematode, less damage and also less buildup. It's like planting peanuts almost in a root knot field as far as the next time you plant cotton. Another thing to point out though is these ones from Phytogen have bacterial blight resistance. The ones from Stoneville and Delta Pine do not. Okay? How important? We'll talk about bacterial blight. I have to talk about bacterial blight. There were some fields in southwest Georgia where bacterial blight was a concern. For most of the state, in fact, I couldn't take you to a field where I feel it was really bad. I'm sure there's people here who could, but for the most part it wasn't. But you get that one chance. Depending upon the variety you plant, you either have root knot nematode resistance or you don't. You either have bacterial blight resistance or you don't. Preaching to the choir, left-hand side, that's what roots look like when you don't use the resistance. Right-hand side is where you do use the resistance. Okay? Well, Bob, we're not growing roots, we're growing lint, right? I understand that. But you have to have good root system to make lint. Okay? Packard Park 2019, that's around close to the Sunbelt Ag Expo. On the left-hand side, Stoneville 5600, Delta Pine 1747, Phytogen 480. That's the amount of root knot nematode buildup at the end of the season. Right-hand side, 1646 with and without 5 pounds of AgLogic. Okay? Well, we're really growing lint, Bob. I understand that. It's important, but I know we're growing lint. Okay? This is part of the value. The nematode buildup means that the next time you go in, not only are you protecting the roots this year, but what happens next year. And in this situation, now you could accuse me of cherry picking, Bob. You're only taking the data you like. Okay? Well, wait. Okay? But in this situation, and this field with heavy root knot nematode population, 1646, when you added the AgLogic to it, and this was seed cotton, you made 100 pounds more seed cotton with the 5 pounds of AgLogic. But you can see the difference when you had the root knot nematode resistant varieties out there. Okay? The point is, does that happen always? Can every grower get that every time? No. But the more your root knot nematode population is, the higher root knot nematode population, the greater chance you are to see these differentials. Okay? Look at the same field. Now this was a variety trial in Colquitt County. You can see the root knot nematode buildup. There's a 1646, there's a 2038. You can see Phytogen varieties, you can see a reduction in nematodes. Okay? No surprise there. All right? When we look at gall ratings, the damage to the roots, no surprise. We get less damage when we use a resistant variety. Preaching to the choir. And this is a situation where we made more cotton with the 1646 and the 2038 than we did with the resistant varieties. The point of this is we don't always make more yield. We always reduce nematode populations from a susceptible variety. We always have less damage, assuming there's nematodes there. We don't always make more yield. When do we make more yield? For sure, we make more yield as your populations increase. But I'm not an economist. But if I was, the other factor we have to put in here, if we're using nematicides, is what's the difference in yield, right? And what's the cost that it took to produce that yield using a nematicide? Okay? Questions so far? Let's talk about two new varieties. 443 in specific, because that's the variety I'm using more and more in the south, or in this part of the state. 443 is what our growers have been using, and 2141 from Delta Pine. 443 from Phytogen, 2141. Okay? And the thing about these is they have both root knot nematode and reniform resistance. And this is 443 last year in the panhandle of Florida, where they had high reniform nematode populations. That's a susceptible variety. You can see the difference in growth. All right? Most of what I test is against root knot nematode. What I have seen, though, is we do reduce nematode populations in the reniform using 443 or 2141. We just don't reduce them as much as we do with root knot nematodes. With root knot nematode resistant varieties, we almost eliminate an increase in nematodes. When we talk about 2141 or 443, I would say we drop the nematode populations reniform by about 2/3. We don't get the same level. Yeah, Mark.
I think I understood you right. Going back.
I'm glad somebody did this morning.
Are you saying that the AgLogic plus a higher yielding variety is still not as good as a resistant variety? Did I understand you say that?
It's not what I said, it's what the data said. All right? Now, this doesn't always happen. I didn't cherry pick, I just grabbed trials, okay? But in this situation, 1646 plus 5 pounds of AgLogic versus 5600, 1747, or 480. Under those conditions, because a lot of growers, I'll be honest, a lot of growers are wanting to be able to use a nematicide with the variety. They want to pick the variety they want to plant and then protect it with a nematicide. Here's a situation where even with 5 pounds of AgLogic, it did not equal what the resistant varieties would do. Okay? Doesn't always happen. But what you need to do is factor in the cost of the nematicide treatment plus the increase in yield. Here it was 100 pounds of seed cotton, and compare that to what you made where you didn't have to use nematicide. We cannot always improve upon the yield enough with a nematicide to equal resistant variety. But there's times where even in the absence of a nematicide, a high-yielding variety may outperform a resistant variety. Depends upon the populations and those things. Yes, sir?
Would I be correct in assuming, though, that over time, your numbers are going to be higher for the resistant variety? Your lint production will be high. I mean, over time. I mean, these are specific trials. But, I mean, you've been doing this how many years?
A long time.
A long time.
Long time to forget how to tie my tie this morning. A long time. So.
But you do remember the numbers for
yes.
nematicide resistance?
Yeah. What I will say, great question. All right. What I can tell you is this: resistant varieties in a non-nematode environment, right, which we typically look to plant, the difference in yield potential between our high-yielding susceptible varieties and our resistant varieties, the gap is becoming smaller and smaller versus what it was when we first brought resistant varieties out. Okay? So I can't say, it takes a whole bunch of data. I won't say that every time you plant a resistant variety in a nematode field, it's going to outperform our susceptible varieties, with or without the nematicide. What I am going to say is the potential, the opportunity for doing that is increasingly on their favor. The yield potential in our resistant varieties is improving and continues to improve. Okay? To the point that I would not hesitate to plant. Yeah.
Have you ever seen a situation that warranted a resistant variety and a nematicide?
Great question. Have I ever seen a situation? Hey, Kale, how you doing? Have I ever seen a situation where you could get a resistant variety with a nematicide? I have seen trials where we have put Telone out with resistant varieties, and we have increased yield a little bit. A little bit. No. Okay? So to say, if the question is, can we improve yield using a nematicide like Telone with a resistant variety, the answer is yes. And I'm not convinced that it's all nematode control. I think that Telone may bring some other things to the table. All right? The other question though is it economically viable? And the answer is it has not been. Can we improve yield? My expectation is under the right conditions, 1646 plus 3 gallons of Telone is going to bring us 400, 500 pounds of lint, right? Under the right conditions. That will pay for itself. Under a variety like 2141 or 443, I would expect a marginal lint increase. Maybe not related to nematodes, maybe related to other ecology out there, but it will not necessarily pay for itself. So the short answer is I wouldn't recommend it. If you plant a resistant variety, I would not use nematicide. Okay? If you're not going to plant a resistant variety, and you're not going to use, and you're going to plant into a nematode-infested field, it doesn't matter if it's root knot, it's reniform, it's sting, it's Columbia lance. If you do not use a nematicide at planting time, you've lost that opportunity. You cannot go back and do that, right? You cannot go back and do that. And so here is the slate for 2022 on what we have. Telone we continue to use. Telone, I don't know about the supply right now. Telone remains to be our best nematicide. Since we lost Timic, Vellum has been out there. Okay, we can talk about seed treatments like Evicta and BioST. Seed treatments are available. They're convenient. If you think you might have a nematode problem, but you're not really sure and you want to put something out there, that might be the answer for you. If you think you have a nematode problem and you got good reason to believe it from nematode counts or damage or a consultant or an agent or seed supplier, I don't think this is enough anymore. Okay? This is where we talk about using Vellum or AgLogic. Okay? Now AgLogic or Aldicarb, Timic went away for a long time. It's come back now, right? And they think that they thought like the field of dreams. You build it, they'll come, right? Well, in the interim between Timic and AgLogic, Vellum came on the scene. And a lot of growers are using that effectively now, okay? But AgLogic is back, 5 to 7 pounds. And it remains gypsum formulation, but it remains a very important contribution for your nematode control, okay? If you're not going to use Telone, you may not get the yields you want, but at least consider using what the AgLogic or the Vellum will do. Okay? We've also got Vidate. Return XL is a generic formulation of the Vidate. We got those that can be used as well. Okay? Philip Roberts would tell you if he was in the room, use Vidate carefully because Vidate will affect beneficial insects. Okay? Just remember going into 2022, the big change happened last year. You may be used to Vellum Total, which came with fluopyram plus imidacloprid. The new formulation, simply Vellum from Bear Crop Science. If you're going to use it for nematode control at 6 to 6.5 ounces, remember there's no imidacloprid in it. You've got to put something with it. On the other side is if you're using Vellum Total, you got some left over, your distributor's got some left over, and you only put the 6.5 ounce rate out there, you're not putting enough. Okay, make sure what product you have. Questions on that? Okay? Show you a little bit of data. This is from 2019, 20, and 21. Resistant varieties. It changed over the years. 5600, 1747, and 480. 2020, it was 5600, 1747, 400, 480, and 443. And last year, that's the data I'll probably show, the 2141 and the 443. It's against 1646. I mean, there's no hiding it. 1646 is a very popular high-yielding variety. So 1646 by itself and with nematicides versus these resistant varieties, okay? And I think that's the question, Mark, you were asking, a couple others were asking. This is 2020 in Colquitt County. This is late season gall ratings, all right? In green, these are the root knot nematode resistant varieties. These are the gall ratings. Yellow is 1646 plus Telone, 1646 by itself, 1646 plus 5 pounds of AgLogic, 1646 plus Vellum, 1646 plus Propulse. And what you can see is two things. As the level of performance in your nematicide increased, we're seeing at least a numeric drop in the amount of damage. Okay, so Telone gave us the lowest. Vellum, and it wasn't a real high environment, so it wasn't that much difference. But you can see 1646, Telone, Vellum, AgLogic. And green is where we used a resistant variety. Okay? When we look at late season nematode counts, this is the resistant varieties to root knot nematode. Those are where we use 1646. The red bar is our economic threshold level. So what this says at the end of the season, we're looking at a situation where if you did not use 1646, in this instance the telone actually did reduce nematodes. It doesn't always happen that way, it usually does not. But if you did not use, if you used 1646, your nematode populations were above threshold at the end of the season. If you planted resistant varieties, they were below. That's great, Bob, but we're not selling nematodes, we're not selling healthy roots, we're selling lint, okay?
Bob, I got a question about that. Your vellum total on the other slide had a much lower
The gall rate,
rating,
I've got the same question.
but
Okay.
it's a lot higher. So is that the longevity of the product in the field, or what's attributed to that?
That's a variation doing nematode work, okay? So we cannot always take the slides, buy them as individual. They sometimes mirror each other well. In this situation, the vellum had a lower, okay? Do I believe vellum is good as telone? It's not, right? But in this situation, the take-home message, rather than making real strong comparisons between one versus the other, it's the fact that if we used a nematicide, whether it's telone, AgLogic, or vellum, we were improving nematode root control, damage control, over if we didn't, okay? One trial, I can't make that kind of a say. If we took all the data you're saying over time, telone would be much better, vellum would be here, AgLogic would be somewhere between telone and vellum, okay? The main point is that at the end of the season, unless we were using a root knot nematode resistant variety, and in this situation the telone helped us too, it usually does not, we're building nematode populations above our threshold level. Okay? Now when we look at yield, and this is what happens when growers a lot of times look at yield, they say, well, look at that, Bob. And again, we didn't get as much performance out of the 1646 plus telone from the, as far as yield bump as we usually get. What does that mean? Maybe the population wasn't high enough to really see a strong value in telone.
Have you got plenty of rain?
If we got too much, this was 2020. 2021 we saw the same thing, and that was too much rain on the telone and the AgLogic. That's why I didn't show that data right here, because the rain affected the performance of the telone and the AgLogic. Okay? The main thing is we did not always, the 5600, the 1747, we didn't always make the highest yields with the root knot nematode resistant varieties. 443, root knot nematode reniform resistant, not only had the highest yield, but we didn't have to put a nematicide underneath it. Okay? Point is, before you say, okay, as a grower, say, well, let's see, 1747 made 804. How much did it cost to make 955 with vellum plus 1646? So it's not just the yield, it's the cost of making it. Okay? Questions? Questions?
It seems like to me, to simplify, like in a 3-year rotation, 2 years of cotton, that second year, you'd be pretty, most of the time, you'd be, to plant an NR variety, you'd be on top. In the second year. Because you get a reduction from the peanuts the first year.
Absolutely.
And then the second year, you got the buildup.
If you plant peanuts, that knocks them back. Then if you plant the nematode resistant variety, that knocks them back. When you come into that third year, that second year of cotton, that third year, then your populations could be very low. Okay? To the point where in the past you might not have been able to grow cotton without the resistant variety telone. You may be down to wear a seed treatment or something like that. But what his question, or what his point is, can you reduce nematode populations enough with peanuts and with a nematode resistant variety that you've really got it good that third year? Yes, possibly, probably, but what's the key? What's the key step there? A nematode sample at the end in the fall of that first cotton crop, just to see what's happening.
That first year after peanuts, you could not plant an NR variety maybe because you've lowered your
Oh, I got you.
nematode. So you get to plant one of the other varieties. So then the second year you have to go back to it.
It's a plan that makes sense, but there's only one way to really know for sure, and that is to take nematode samples and see. But a year out of peanuts into cotton, certainly that's going to help you. But I can promise you, does a year of peanuts eliminate a nematode problem in a cotton field? It does not. Because if it did, we wouldn't have to worry about nematicides, right? Because a lot of people do that. It helps a bunch. Bacterial blight, just really one slide on this. Bacterial blight was not a problem across most of Georgia. In fact, for me, it was not a problem. But I talked to consultants in Bainbridge yesterday, and they said, well, in this field or that field or this field, we really think we lost some yield. What was the common denominator? Southwest Georgia, buku rain, and a susceptible variety. Okay? If you have no stomach for that, and who does, right? I can tell you most of us did not have a problem, but if you don't, you have the two choices. Plant peanuts or plant a bacterial blight resistant variety. Okay? Questions on that? And this is just showing some of the varieties coming out with bacterial blight resistance. Some of the other ones that have bacterial high-level resistance, mid-level resistance, and I would encourage you as we get new varieties coming out in 2022 available to you, at least educate yourself on which ones have better resistance, which ones don't. Okay? Questions on that? Just a little bit on Fusarium wilt while we're talking nematodes. Tucker, in your county, Fusarium wilt is a significant problem. In fact, in my world of cotton, it's very restricted. It's restricted to areas typically where we have sting nematodes, but it can occur where you have root knot and reniform nematodes. It's typically restricted, marked to your area of the world, southeast Georgia, a little bit of southwest Georgia. But I've seen it as far north as Bishop, up and around Athens, Clark County area. Most important thing to note on this is if it's in the field, it is a tremendous problem. But it's always in Georgia, always associated with nematodes. Texas, that's not the case, but in Georgia, always associated. The way you diagnose it, you're looking for this tiger striping, this intervenochlorosis. This can be a fertility problem, this can be a nematode problem, but the way you take that and split that lower stem, you will see, if you have that discoloration, it's going to be, the vascular discoloration is Fusarium wilt. Okay? Another important thing. 2000, when I started, you showed me that picture, I was going to say that's Rhizoc. That's Rhizoc, right? It's seedling disease. But you look and you see these marginal edge necrosis, and you see the yellowing. Even seedlings, you can take those out in severe cases. In the Fusarium wilt fields we have, you split those lower stem and you're going to see that discoloration. That's Fusarium wilt as well. Why does it matter? The main reason it matters is at this point, what can you do about Fusarium wilt? That train's left the station. There is nothing you can do about it, but you can do something for next year. Because the easiest way to move this, to spread this, is on equipment. And if you move tractors and equipment into a field like this, and you take the dirt from that field into a clean field, you will spread the pathogen. And if the nematode is already there, you will spread it. So the main thing is to be careful not spreading it. Okay? How do we manage it? You get that one chance. We have to manage the nematodes. We have to manage the nematodes. Tatnall County on the left-hand side, that was vellum. Vellum did very well in that situation. Chris Goodman here in Tift County, unless he puts telone out to control the nematodes, he cannot control it. Yeah, Hunt. Great question. You beat me to the punch. Okay? If you have a root knot nematode problem in a field where you have Fusarium wilt, you should get benefit out of the resistant varieties. If you have reniform nematodes in a Fusarium wilt field, you should get benefit. The problem is this. What's most of the nematodes that affect our Fusarium wilt? Sting nematode. We just don't have resistance. The other thing is, depending upon how the resistance work, Hunt, if the resistance works by keeping the female from producing giant cells and galls, but does not prevent her from punching holes in the wall, you could still be opening a door for the Fusarium to get in. So in theory, resistant varieties will be a help. I would encourage you to use it, but you got to make sure, A, that it's the right nematode. If it's sting, it doesn't matter what you plant, you need a nematicide. Second thing is to understand how the genetics are working in the resistance. Okay? Great question. Target spot. Okay? Target spot last year was a good year for target spot, a bad year for growers. Too much rain, too much splash, too much growth, too much coverage. Okay? What I can tell you is that we now have, going into 2022, I have 3 fungicides I generally talk about. We got azoxystrobin. Azoxystrobin is not very strong on target spot, but it helps. Our best materials are Preaxor from BASF and Miravis Top from Syngenta. Those products are the most effective at target spot. Yesterday in Bainbridge, a consultant told me that he sprays every year because he believes they're going to get a benefit. They're going to spray the 3rd week of bloom because where they are, they don't get any break from target spot. They're going to spray. My suggestion for anybody who doesn't have that kind of pressure is before you treat for target spot, as you approach first bloom, go out there and look and see. Do I have it developing? If it's not developing, I would hold off on the fungicide application. That's one cost we can reduce. Use it if you need it. If you don't need it, don't use it. What's my biggest concern for fungicides going into 2022? Two things. Cost is going to be up, but so is everything else, right? This one, which has been the workhorse for target spot, is going to be in very, very tight supply. So you can see if you're going to be able to get it. If you can't, Miravis Top may be available. Azoxystrobin, whatever formulation it is, it's available. It'll be more expensive. It's less effective. Let's talk about this disease. Okay? What's the method of my madness? I've thrown a lot of information at you. I don't expect you to remember all of it. All I expect you to remember, I hope you remember, there's 7 points. Linearia, nematodes, bacterial blight, Fusarium wilt, target spot, and wait, there's more. Number 6, areola mildew. Okay? Stephanie in the back has done some trials when she was still a county agent in Brooks County. All right? Areola mildew is something that used to stay home in southeast Georgia, over around Appling County, over there around some of the Ware County, okay, Bryan County. But since about 2017, and I can't tell you why it is, it's coming earlier. It's even getting into some of the southeast states like Mississippi and Alabama as well. Okay? This is a disease which is unforgiving. This is a disease which you need to take seriously. The survival goes through several stages. It's surviving in that crop debris we have. The life cycle means that if we got it out there in the debris, it'll probably be surviving. And the warmer it is, the more it's likely to survive in debris. Okay? The symptoms, very much like target spot, early or very, very severe premature defoliation. It has a frosted, very distinctive look on those leaves. As it affects the leaf, the frosting, the spores are there, and it crumples the leaves up. You can see that. What do we do about it? If you are within 4 weeks of defoliating your cotton, I would not worry about it. For 2 reasons. If you're within 4 weeks of defoliation of your cotton, you're not going to have enough time. The disease does not have enough time to cause damage, or it's already caused so much damage it doesn't matter anymore. Okay? Either it doesn't matter because it's too late, or it's too late for you to do anything. But if you're with further than 4 weeks out, I'd pay attention. Is it you who says the 5th week of bloom is what you look at? Okay? A lot of our agents, I don't know about consultants as well, looking, but 5th week of bloom, between the 3rd and 5th is a critical period of time for that. Okay? This is from last year from Colquitt County. Azoxystrobin. The good news for areola mildew is azoxystrobin is more effective than it is on target spot. Preaxor and Miravis Top. 3 rating dates, 37 days after treatment, 51 days, 76 days. The yellow bar is the defoliation if we don't treat at all. The red bar is if we use the azoxystrobin. The green bar is if we use the Miravis Top, and the purple bar is the Preaxor. By the end of the season, you may not be able to tell much difference. But within 37 up to 60 days after application, we are seeing a benefit from a single application of fungicide made at the 5th week of bloom. Okay? When you look at yield, in this situation, it looks like there's not much yield. Statistically, there was, but we see the same. This is one of the few where it's not statistical. We see this every time we treat. Stephanie saw it. Seth saw it. Bill Tyson in Bullock County saw it. 100 pounds of lint, approximately between 100 and 250 pounds of lint. Okay? This is from Bullock County last year. It's from Bullock County to Terrell County to Brooks County to Colquitt County. This was where he didn't treat. This is where they used tebuconazole, which is labeled, but you can see there wasn't much difference. Miravis Top, Preaxor, and the azoxystrobin. You can see the impact. This is all areola mildew in that field. That's what happens. Okay? Why is this white and that's not? Because we still got leaves here and we don't here. When we look at yield, that is lint. Okay? And you can see where the Miravis Top, we made 1,500 pounds of lint. The non-treated made 1,200. About 300 pounds different in lint from treating. Okay? A little bit hard to see, but Seth did a trial last year where he was looking at aerial application. This was ground equipment. Seth is with aerial application. Aerial mildew. He looked at 2 gallons per acre, 5 gallons per acre, and not treated. Percent defoliation on that day with the aerial mildew, percent defoliation was 25% versus 2 and 3%. It does make a difference. You get better control with 5 gallons. You still get adequate control with the 2 gallons. And I don't have the yield on here. What was your yield benefit? The difference between 5 gallons, 2 gallons, and 0 gallons.
100 pounds difference each treatment.
So 200-pound increase with 5 gallons versus not treating, and 100-pound increase with 2 gallons versus not treating. So it's real. Terrell County, Bullock County, Colquitt County, Brooks County. I'm going to stop there.
Dr.
Okay?
Kemerait, I got a question.
Yeah.
In regards to the areola, along with target spots. I know you've been very
You got sludge,
passionate
put it there.
over the last month about cold weather.
When he says I've been passionate, I'm always worried. What did I say? What's he going to call me?
Know that
Okay,
we
so
need
yeah,
cold
okay,
weather.
you're right.
So in referencing, areola or target spot, and the inoculum that's left over, where do you see a break in the amount of inoculum versus cold temperature?
Great question. For the nematodes, his question is, where's the break? Where's the reset button?
Right.
All right? For nematodes, the reset button is colder temperatures, keeping those soils cold, killing back the debris. The number one thing that's going to help us with target spot and aerial mildew is not the temperature. Temperature is second. Rainfall is going to help us. Anything, that aerial mildew, that target spot is going to be much more severe if we don't break that material down. Rain is going to help us to rot that. We deprive it of a food source. You deprive them of a place to survive. So right now, it's the rain more important than the temperatures as far as the aerial mildew. That aerial mildew, that tar spot, that target spot, it will exist on crop debris. Chokes me up, think about it. Anything we can do to either bury crop debris or break down crop debris will reduce the threat. Okay? I'm going to ask for the last few minutes here, I'm going to ask Dr. Sudeep Bag to talk to you because he is one of the leading experts in the state, or he is the expert in the state and in the country in cotton leaf bollworm virus. This is a disease which since 2017 we've been chasing, and I think he's going to give you the information we need. Remember, anybody in here, thanks for the patience with me as far as going first, not Camp being in here. If you need the pesticide credits, go out and go buy the kiosk there. Out and on to your right, they will have pesticide credit forms for you. Okay? All right, Dr. Bag.
Thank you, Dr. Kemerait, for the time, and thank you, everyone. I will just take few minutes so that you don't forget there is one more disease that we need to care about or that we need to keep remembering, and that is cotton leaf roll Dwarf virus. As Dr. Kemerait mentioned, it was first detected in Gulf Coast in Alabama, and in 2019 we are able to confirm its presence in Georgia. So since then we are working on it, and I will just give a reminder what all we have learned so far, and these are some of the words that we know, and I don't want to explain as most of us know about these words, what these mean in case of viruses. But over the period of last 3 years, we looked into different symptoms and we tried to see if there is any variant in the virus that is causing those symptoms. So we looked at symptoms that were causing early in the season from 3 different counties as well as late in the season, and what we see if there is any genomic difference in the virus. So these are the symptoms that we collected from early season, and these are symptoms that we see in late in the season. Early symptoms, when I say, those are reddening of the leaves, turning down of the leaves, petioles are red, whereas late in the season that we observe after August, that is stalk internodes that are stacking or crumpling of the leaf deformation. Those are the symptoms that we observe after August. Early is the redness of the leaves. So we looked at these different symptoms, and what we see based on the sequences is that what all isolate what we have in Georgia, they are all similar, irrespective of the symptom, irrespective of location across Georgia. Over last 3 years, what all collection we have done, based on last 3-year process, we sequenced lot of another isolates this year to see if there is any change happened over 3 years. We are not seeing any change happening over last 3 years. These are different weeds. So where this virus or the vector, in this case aphid, this virus is transmitted by cotton aphid. So where this aphid or this virus is surviving once the cotton is not there in the field. So we looked into different weeds that are found in and around cotton, and this is over the period of time during growing season and when the cotton is not there. So we found that across 23 different weeds that are showing positive, the virus is present in that one. And these are some of the weeds where we have 34 different weeds where we have not detected the virus. That doesn't mean that they are not carrier. At that point, we have not detected the virus. As Dr. Kemerait mentioned earlier, this particular stocks, if it remains and if we are not killing it due to the hard winter and in the little bit of warmer temperature, the green leaves are coming up. Those leaves are carrying the virus, and those leaves are the one where the aphid will feed, and that will give the initial inoculum for the virus to proceed to the next crop. So we need to take care of this stock that Dr. Kemerait was earlier mentioning. Going back to 2021, what we have observed, we have not observed any significant yield loss or any big grower commercial field where we can say that there is this virus issue due to which we are having any yield loss. So I will just go through some of the symptoms that we have observed. In this case, this is on July 21 where we see this type of symptoms, and almost all the field, we tested like 20 plants in this field, and almost all the plants were showing similar symptom and all are carrying the virus. This is in Sumter County. This is the symptom what I am talking about earlier, where we see the leaves drooping and then there is no ball in this plant. So if the virus is there, there are some impact on the individual plants. This is what I see in the Colquitt County, where the plants are way higher, like 40, 42 internodes. And what we see in some of the cases, there is no bolls above 15 to on the top internodes. Below 15 internodes, there are some bolls or some cotton remains, but above there is nothing. And these are some of the cases where we see in the infected versus in the other one that may not have the virus.
Dr. Sudeep, go back one slide, the bronzing. Since there's no test for bronze wilt, there is a PCR test for cotton leaf roll dwarf virus. How are you going to differentiate between the two when you see that?
What is
because we know we got a couple varieties that, you know, kind of look like bronze wilt, and there's been, you know, chattered that it is bronze wilt in a couple varieties.
What this question is asked since we started working on this virus, whether the bronze wilt and virus is the same, or can you differentiate between a bronze wilt and the virus? So far, I don't have anyone told me that, Sudeep, this is bronze wilt, this is virus, and let me know if you find virus on this or not. Give me a plant, I will try and I will give the answer. So far, I have not having that answer, but 99% of the plant that we have collected with this symptom are turning positive or the virus is detected. So the question I will raise is bronze wilt is caused by virus, because there is bronze wilt is no pathogen is associated. It's a physiological disease or symptom that was told to me that, okay, this is some sort of physiology, but no pathogen is associated. right?
We did not look, I can tell you back then, we did not go to the same effort to look for a virus in it that we do now.
So, I mean, back in the paymaster days, it could have very well still been this because, I mean, I've been in the cotton field for 25 years. I see plants like that all the time.
And we weren't looking for it
Right,
as intensively as
right.
we are now like that.
So if you have any sample from 20 years back, give it to me, I will look into it.
That's what I thought you were going to say.
Okay, so these are some of the symptoms that we have observed in Colquitt County, and this is what we saw in our trial in Tifton. This is planted on June 2nd, harvested on November 8th, and on 90 days I saw this symptom of the wilting. And I tagged those plants, and this is at the harvest time what that plant was looking like, and this was asymptomatic, and what was the yield. So there is the difference when the individual plant is infected and there is no infection.
3 more minutes.
Yes, so this is another one that we have observed in our Tifton, where we see this symptom we observed in August, and then I tagged those plants in August, and after that you see there is nothing. So up till here, that is 15-16 internode, we have some crop, some yield, but above that there is nothing. So we took this plant and what to see whether we have the virus or not. So this is how the virus look like. It is like 23 nanometer, and this is what the virus look like, these small arrows. This white one, this is how the virus look like in that particular plant. So this is the additional evidence apart from PCR test. Yes, the virus is there. This is, I have not done it from that red leaves. This is how overall, if I say on the infected plant, individual plant, where we in the symptomatic one, in 4 plants what we have, and in asymptomatic what we have the yield. Last year we tested around 4,700 sample. Of that 26% are positive. That is all from the growers field and the other total research project. In our 16 out of 17 counties, we have tested positive for the presence of the virus. Over last 3 years since I am here, the virus was detected in almost all the cotton growing belt in Georgia. These are some of the other works what we are still trying to pursue and trying to understand how this virus is playing, causing disease in our crops. And these are some of the publications that I have that are always available, and feel free to reach out if you need any further information. And these are some of the another symptoms. Again, Dr. Roberts, see the plant height here. You remember this? This is again the other one in Colquitt County. See the height of the plant. This is in 2020, this is 2021. 100% plant are showing symptom and virus is detected. This is 2020, see the height. There is Dr. Bob Kemerait, and here is Dr. Bob Kemerait. See the height of this one versus this one. So this is 100% of the plants were infected, showing
Question for
symptom.
Dr. Bag.
So the symptom is highly variable across the state, across the variety, and depending upon the lot of other factors, the symptom can vary. But this is all I have,
Bull
and
rot was
thank you
huge last
everyone
year.
for your
So
support.
the most important thing we can do is we can basically eliminate bacterial bull rot, but that's not the main one, okay? In a year like we had last year, with the amount of rain and the splash that's coming, is very, very difficult. We had hoped, if you look at, I think that Dr. Hand will show some of his data with wide row or skip row plantings. We're hoping anything we can do to open the canopy up will help us with that. And his data went backwards. We weren't seeing any benefit. But what I can tell you is, the most common one, Mark, and this isn't what you want to hear, anything we can do to manage growth, to keep the growth and not rank, that helps. What we're hoping, what we're testing, and you say, well, I've heard all that, Bob. You've been saying that since 2000. The one thing that is changing is some of these fungicides, like the Mirabis top, are extremely aggressive on fungi. And what we're hoping is there's at least some potential that even though we don't get the concentration we might want when we treat for target spot aerial mildew, there may be some benefit that we haven't seen before because of the improved quality of our fungicides.
That'd be a game changer.
It would be, and remember, I said if, right? I used the word if, but I will say there's some hope that our fungicides are improving to the point that we may be able to get better. But as far as right now, the reason we don't talk about it more is because we don't have good answers for it.